Hi everybody, in this brief video we'll go through the slides that we couldn't cover yesterday, and I wanted to basically go over what different types of transgenic animals we can create by introducing different types of transgenes into their genome. So far we discussed how you make a transgenic animal, and now depending on what is that you're introducing into the genome, the type of animal that you get. So a regular transgenic is simply having a foreign sequence introduced into the genome. Typically what we're trying to do is express genes that are not normally expressed by the host species, in this case a mouse. So if you simply have a regular promoter and some ectopic sequence to be expressed, we call those regular transgenics. You have some new cDNA under some promoter, and that is what is inserted into the genome. When we want to make a knockout animal, what we simply do is we target the gene that we would like to knock out from the mouse genome, and we don't introduce anything in its place. So we're knocking out the gene from where it was normally, and then we're replacing it with something that is inert. In this case, it would simply be the selection marker that we use for the process of making the transgenic animal, let's say neomycin resistance gene. You simply put a neomycin resistance gene where a gene that you wanted to knock out normally was. And if you follow the steps of making that transgenic and you get all the way to making a homozygote transgenic for neomycin resistant, essentially what you've done is replace the endogenous gene for this neomycin resistant cassette, effectively taking out the endogenous gene from the mouse genome. That is shown here on this slide. So if you proceed with the process of making homozygote transgenic, but instead of introducing a gene of interest, you simply introduce neomycin resistance, what you've effectively done is take out a gene that you wanted to knock out from the animal. So instead of asking what are our homozygote transgenic animals, essentially what you're asking is which ones among your progeny of the last cross are your knockouts. And in this case, assuming that the neomycin resistant would make the original gene shorter than it was, you would be looking for those animals in which you only get the small PCR product, indicating that the original gene is gone from the genome. In some cases, we may want to overexpress a foreign gene. It's not only about introducing a gene that is not normally in the mouse, but it may be a gene that exists in the mouse, but we want to make a lot more of it. In those cases, what we do is we introduce our cDNA of interest, new or old, and by that I mean whether it exists in the mouse or not, and then we put it under a very strong promoter. In class, we discussed briefly the use of viral promoters, for instance, to drive very high levels of expression in eukaryotic cells, and that's certainly something you can do. So you introduce a sequence to be expressed downstream of a very, very strong, usually viral promoter. Sometimes you may want to express a gene, again, new or old, in a place where it normally isn't. You may want to drive some ectopic expression of a gene. So the difference here is your cDNA to be expressed is again the same as before. It may exist in the mouse already or not, and the difference in these animals is that you're using a tissue-specific promoter. These are collections of DNA binding sites that will be bound by transcription factors only in certain tissues, a tissue-specific promoter. And then whatever sequence you'd like to express. That's how we can start expressing, let's say, a gene that is normally expressed in neurons somewhere else, in mammary glands, in the intestine, in muscles, and then ask the questions that we want to ask. Other times, what we may want to control is not so much what we are expressing or where we are expressing it, but when we are expressing it. So there may be research questions that have to do with what happens when a certain gene is expressed now. And for that purpose, what we do is we use inducible promoters. So these are promoters that will respond to transcription factors that bind to DNA or not, depending on a certain condition. Think, for instance, about those repressors, inducible repressors that operated in the bacterial operants that we could make bind DNA or not, depending on the presence of lactose or tryptophan or what have you. So there are systems like those in eukaryotes as well, and there exist promoters that will be induced, or in other words, that the factors that bind them will actually bind DNA under certain conditions. Those include heat shock promoters. So these are promoters that respond to transcription factors that change in their shape when it's a little hotter. So whenever there is a heat shock to the animal, these DNA binding proteins will bind DNA and induce transcription. There's copper responsive inducible promoters. These are transcription factors that, as the name indicates, respond to the binding of copper ions. There is doxycycline or tetracycline inducible promoters. So it's a whole series of them. And in the lab, in a test tube, we can hook these promoters to any given gene that we would like to express in an inducible manner in the animal. And if we put the whole thing into a transgenic mouse, now we have a mouse in which we can induce the expression of these genes of interest. Then there is this very special kind of transgenics. We call them reporters of gene expression. And in these animals, what we do is we introduce foreign DNA that includes an endogenous promoter. So that means the natural promoter for any given gene that we're interested in. But instead of having the sequence for that gene, the sequence that is normally encoded in that gene, we hook that endogenous promoter to a reporter gene. So it could be a fluorescent protein, that's what we mean by FP there, or it could be the sequence for LAG-Z, because once a cell is expressing beta-galactosidase, we can reveal the expression of beta-galactosidase using that XGAL compound that will turn blue when beta-galactosidase cleaves it. What you want is a protein that can reveal its presence to you, typically via microscopy, under the control of an endogenous promoter. So essentially what you're doing is you're expressing a protein that you can see under the instructions that a natural gene would normally follow. When you make a transgenic like that, you can end up with things like this. In this case, fish that express a fluorescent protein in certain cell types, and you can follow that simply by looking at where the expression is coming from, or these mouse embryos, I believe they're mouse embryos, that express beta-galactosidase, the LAG-Z gene, in what appears to be, in this case, the neural cord, I believe, and other neural tissues. So the point is that you're hooking either a fluorescent protein or some other reporter downstream of an endogenous promoter. And you're not using these animals necessarily to understand what happens when a gene is expressed in a given tissue or at a given time, but instead to find out how and when an endogenous gene is expressed in the animal. Another version of the same concept is that of tagged genes or tagged transgenics. And the idea here is you have your endogenous promoter, so that will express your transgene of interest where it normally would, right? It would follow the normal instructions for expression. And then you hook that endogenous promoter to a tagged cDNA. And what that means is we hook the sequence of nucleotides that would express green fluorescent protein or some other fluorescent protein to an endogenous gene, such that when that tagged gene is expressed in cells, you would get expression of the protein as usual, and then a few extra amino acids that will make that, in technical jargon, we call it the fluorescent moiety. It's simply a little bit of protein that is added to your natural gene that will maintain the capacity to fluoresce under the right excitation wavelength. So instead of having your regular wild-type gene giving rise to the regular protein that the animal would normally express, you're expressing a tagged protein that you can follow. And the advantage of that is that now you can see where the natural protein would go. With the reporter transgenics, we follow expression at the level of transcription. Anywhere a gene would have been transcribed, you get GFP being transcribed and translated, so you see where those cells were. But in this case, you're actually following where the natural protein is going by following the fluorescence. So here are simply examples of that. These images show how these different tagged proteins ended up in different places within the cells. In this case, they must have ended somewhere in the cytoskeleton, and this appears to be the rough endoplasmic reticulum, and those are some type of vesicles inside a cell. So you can follow where a given protein naturally goes by simply following the fluorescence within the cells. And finally, we have knockdown transgenics. This is something that we do when knocking out a gene entirely is a little too much for the animal, and the animal can't take it. So instead, what we do is knock down, that is, inhibit the expression of a given gene. And we do that by having a strong promoter that will ensure very high levels of transcription of a very special sequence and microRNA sequence that would have the general structure shown down here. So you would have any sequence X that belongs to a gene of interest, a gene that you would like to knock down its expression or inhibit, some sort of linker sequence. These are typically somewhere between 10 and 12 nucleotides. And then the same sequence that you had before, but in reverse complementary orientation. What that means is that when these foreign DNA gets transcribed at high levels because of that very strong promoter that you used in these transgenic animals, the RNA will fold on itself and engage in an intramolecular base pairing such that you form one of these famous hairpin structures that will be processed by Dicer and will generate these short RNA sequences that we call microRNAs that will be loaded onto the RISC complex and lead to the knockdown or the inhibition of a target mRNA. So if you would like to inhibit the expression of mRNA Z, then what you do is you introduce into the animal some sequence that is specific to gene Z, a linker, and then that same sequence in reverse complementary orientation, and then you let the cells deal with the rest. When these very special constructs get transcribed, they will form the hairpins that will give rise to the microRNAs that will get loaded into the RISC complex and lead to inhibition of the mRNA for gene Z. All right, that's all I had for today. I apologize once again for what happened in class today, the delays with my car travel and the Wi-Fi. I hope that you enjoyed this video, you found it useful, and I will see you next in class.